So hey guys, what's up? So welcome to World News 20 new channel. So let's talk about why do some COVID-19 patients lose their sense of smell. Loss of the sense of smell and taste, one of the more recently identified symptoms of the COVID-19, and is now recognized as such by the World Health Organization and the health authorities of some countries, including the US. A new study in mice has sought to explore whether this symptom appears in some COVID-19 patients. They have reported their findings in the American Chemical Society's journal ACS Chemical Neuroscience, tracking the proteins SRS-CoV-2, the virus that causes COVID-19 hijacks two human proteins to invade the cells. One is the ACE2 in the receptor on the cell surface. It opens the door for the virus, while the other is called TMPRSS2, which the virus used to replicate its genetic material in mice. The researchers found these two proteins are produced by certain cells of the nasal cavity that contribute to the mice sense of smell and others within the olfactory epithelium, which is a tissue lining the nasal cavity that is involved in smell. The sustenicular cells have the highest level of the SRS-CoV-2 receptors. Researcher Rafał Batot of the Nicholas Kopernikus University, Poland, told the news agencies the substenticular cells help transfer odors from air to neurons. Batot and the colleagues first used various methods to measure the levels of the expression of the two proteins. They found that the mature olfactory neurons do not express ACE2, while the substenticular cells do. The sense of smell in COVID-19 patients appear to be lost, but the substenticular cells assist neurons in the sensing outdoors, probably by processing odor-binding proteins. Batot said identifying these cells could help in the development of more accurate diagnostic tests. The researchers said they have called for future studies should examine whether substenticular cells can pass the virus to neurons, which could provide SRS-CoV-2 as a route to infect the brain. Age is a factor. Researchers also found that larger amount of the proteins that are made in older mice than in younger ones. These are significant findings because the more entry proteins the whole cell has, the easier it is for the virus to bind and try to infect the cell. The high levels of the entry proteins in the nasal epithelium may explain why older humans are more likely to become infected with the novel coronavirus than younger humans. But Batot said the question is whether what was observed in mice happens in humans too. It is known that the entry proteins have similar expression patterns among the mammalian species, meaning that the mouse appears to be a suitable model for the human condition. But Batot said why the nose matters. Although research by the news agency April 29 identified the two kinds of cells in the nose as the likely first entry points for the virus. These are the goblet cells, which produce the mucus, and the ciliated cells, which help sweep mucus to the throat so it can be swallowed. The study too had drawn its conclusion from the expression of the two entry proteins using the Human Cell Atlas database. They looked at data from the different tissues of the non-infected people. It found that these two proteins had the highest presence in the goblet and the ciliated cells. This is a key difference in what the two studies looked at. The previous study by the researchers of the UK's Wellcome Sanger Institute looked at the human cells and identified goblet and ciliated cells as the likely first proteins of the entry. These occur in the respiratory epithelium, while the new study looks specifically at the olfactory epithelium. Asked if the findings are in conflict, Batot said that all studies depend on the method said. However, what is sure that it is substenticular cells in the olfactory epithelium at the highest level of the SRS-CoV-2 receptors as compared to the other cells in the olfactory epithelium and ciliary goblet cells. I may say the same as compared to the other cells in the respiratory epithelium, but direct comparison between the respiratory and the olfactory epithelium requires further studies with more techniques. Batot said there are few studies now, and some require further examination to resolve inconsistencies between the results. That is how real science works, I think. So if you like this channel, so kindly subscribe to it.